Hello and welcome to the first FAR seminar of the academic year. Thank you all for joining us. We have a very nice turnout. I just wanted to say a couple things about the FAR. And this is a response to the need to give graduate students, postdocs, and early career astrobiologists chances to talk about their research, share it with others, and get feedback as well. So we are doing this seminar once a month. And in order to make it interdisciplinary and address the areas of interest that this community wanted to hear more about, we put together a whole schedule for the year, and that was done by the Science Organizing Committee. And it's interdisciplinary. It covers areas that they felt was important to be covered, and we have a schedule up through June for topics. We're still lining up speakers, but we do have our speakers lined up for next month, which is, I believe, November 3rd, and it's Jen Eigenbrog and to look up the other name, but you can check the website for next month, and it's on extreme life, so we hope you can join us then for that. And I'm now going to turn it over to Carl Pilcher, who will be introducing our speakers today. Thanks.
Well, good morning or afternoon, everybody. I am really glad that the FAR seminar has started up again. This was something that we had going until a couple of years ago, and then I think it fell victim to the big budget cut we took. And now I am really grateful to Estelle and to everybody who has served on the organizing committee for getting it going again, and the fact that you've got it laid out for most of the next year I think is really fabulous. So giving this kind of both visibility and opportunity to graduate students and young researchers, I think, is really part of what the NAI is all about. So without further ado, I will just go ahead and introduce our two speakers this morning. The first speaker is going to be Catherine Neish from the University of Arizona. Catherine is very close to getting her PhD, and she's been focused on the formation of biological molecules in liquid water environments on places like Titan and what that can tell us about the origin of life on Earth. Catherine is originally from Canada and has a BS from the University of British Columbia in physics and astronomy. Our second speaker is going to be Billy Brazelton from the University of Washington. He's going to be speaking about serpentinization and early life. Billy has a bachelor's degree from the University of Minnesota in genetics and cell biology. And without further ado, Catherine, take it away.
Thanks, Carl. I hope everyone can hear me. And good morning to everyone on the West Coast, and good afternoon to everyone on the East Coast. Today I'm going to tell you a little bit about some of the work I've been doing for my PhD dissertation, trying to figure out some of the big questions about the origin of life and how we can use Titan to tell us about maybe some of the things that happened on early Earth. So I think we're just getting the screen up. Excellent. And I'm going to do that on my own computer. Get my little pointer out. Okay, great. And all right. So one of the big questions that we really want to ask, and I guess do you guys see the next slide come up, or?
We're on your first slide.
Okay, I just moved it, and I don't see it moving. So.
Are you on the computer? Are you on the computer that's logged in as U of H?
If I can get it to move.
Click the arrow key one more time.
I click the arrow key. Okay. Sorry, guys. We've been having some technical difficulties this morning, and now my slides aren't advancing. So,
Catherine, are you on the computer that says U of H? U of A, I mean.
Okay, I'm going to use a different computer now. Did that work?
Which computer? Tell me how you're logged in.
I don't know. UA. Can you see it now?
Yes. Try advancing it now.
Hooray. So the big questions. Are we at the big questions? Yes?
Try advancing it. It's not going. So are you using the computer that says U of A?
I was. Now I'm using the one that says UA.
That says UA. Okay. Hold on. Let me switch it to there.
Sorry.
Okay. We are now on the one that says one of the big questions. Go ahead.
Excellent. Thank you. So the question is, how did life begin on Earth? And so one of the first steps towards making life is you need to make the building blocks of life. And there are a couple ways you can get building blocks of life, either through getting it from exogenous sources, coming from comets and meteorites, or the topic I'm going to focus on in this talk are the in situ processes happening in, say, an ocean, or what I'm going to talk about in atmospheres. And that's, you can kind of see that on the left here. So one of the I'm trying to advance the slide, and it's not advancing. So that's super.
Try hitting it one more time.
Alrighty.
I think it's just catching up.
But so something that Stanley Miller did in the 50s, kind of starting this whole field of prebiotic chemistry, was to take an atmosphere and spark it and see what he could make. And what he used was what's known as a reducing atmosphere. It had a lot of molecules that were willing to give up electrons. And so the topic of this talk is sort of what can we make, what interesting simple organic molecules can we make from reducing atmospheres. And my slides still aren't advancing, so that's great.
I can do it for you. Why don't you just say next slide?
Next slide, please. Excellent. Okay. So we're going to talk about the chemistry of reducing atmospheres. And Titan is one great example of a reducing atmosphere. It's made up of methane and nitrogen. And if it's constantly being bombarded by solar UV photons, as well as charged electrons coming from Saturn's magnetosphere. And what happens is it breaks apart the methane and the nitrogen, and they then recombine to make a whole suite of new and more complex organic molecules, known as hydrocarbons and nitriles. And hydrocarbon is just basically any organic molecule with both hydrogen and carbon in it, and a nitrile is an organic molecule containing a CN functional group, such as hydrogen cyanide. And so just to give you a bit of a schematic about what is going on there, here's a beautiful picture of Titan taken by the Cassini spacecraft. Or there we go. And we start with methane and nitrogen, and then they're bombarded by UV and electrons, and then we get this whole great suite of organic molecules that we've actually observed in Titan's atmosphere through both the Voyager and the Cassini spacecraft. And right at the bottom here, in addition, going back, okay. In addition to these gaseous molecules, we also have a big hazy aerosol, sort of like what you see over Los Angeles on a really bad day. You get this haze of heavy organic muck. So that's Titan. Titan is one example of this reducing atmosphere where we can make all these simple building blocks. The early Earth might have been another example, which is sort of how this ties into the origin of life. There's been some new work that suggests that the early Earth may have been fairly rich in hydrogen, which is a very reducing molecule, and such an atmosphere might have been able to produce molecules like hydrogen cyanide, which are considered key to terrestrial prebiotic chemistry. In addition, it's possible that the early Earth may have had quite a bit of methane in it in order to raise the temperature, because back at the beginning of the solar system, the sun was a lot fainter, so the Earth was a lot cooler, and you might have needed, say, methane and carbon dioxide to increase the temperature. And if you had comparable amounts of methane and nitrogen, you might have formed a haze, not unlike the one that we see on Titan today. So sort of a analog there. Now, okay, so we're making some of the building blocks, these simple organic molecules. But actually making biomolecules, amino acids to make up proteins, or the building blocks of DNA and RNA, that's more difficult to do in an atmosphere. On Titan, the problem is there's really not much oxygen in its atmosphere. And if you want to make a biomolecule, you really need oxygen for most of them. For example, here's the amino acid glycine, and you'll notice there's two oxygens here in red forming this carbonyl group on the side here. So you're going to need oxygen to get a biomolecule, and that's basically lacking in Titan's atmosphere. The early Earth, there was probably quite a bit of oxygen in the form of carbon monoxide, carbon dioxide. However, if you try to make organic molecules out of such an atmosphere by sparking them in sort of this Miller-Urey experiment, you don't actually make very many prebiotic molecules. Miller did some work in 1983 where he took his original experiment and added CO and CO2, and the only amino acid he was able to make was glycine, as opposed to his original experiment done in a reducing atmosphere, which produced quite a few amino acids. So, okay, we've got organic molecules, but we don't have biomolecules. Is there another way? And so what I'm proposing in this talk is that there might be another way. Instead of making them directly in the atmosphere, is if you have those organics made in the atmosphere. Oh, can you go back one? Anyways, if you have those organics, they might rain down onto the surface and interact with liquid water there. And on Titan, there was some work done in the 80s where they took Titan tholins. These are these haze analogs. And I'm going to use the word tholin throughout the talk, but basically what I mean by that are these haze analogs that I'm making in the laboratory. And if you put them in water and hydrolyze them, hydrolysis just refers to any reaction with a chemical compound in water, you will produce amino acids. On the early Earth, it turns out if you have a high enough concentration of hydrogen cyanide, you'll make quite a few biomolecules. Next slide. Things that make up DNA and RNA, as well as amino acids, simply by hydrolyzing HCN oligomer. An oligomer is just a connection of a lot of hydrogen cyanide components, which I have highlighted, but I can't get to that slide. So, okay, I'm just describing what I mean by something called a purine. For example, adenine is a purine that makes up DNA and RNA. Cytosine is a pyrimidine, also very, very important to the information structure of molecules. And then amino acids like glycine, the one I showed you before. So, next slide. And just explaining what an oligomer is for non-chemists. All right, so you can see how we might have liquid water on the early Earth. There is evidence of having liquid water on the early Earth, but if you're familiar with Titan at all, you might think this is crazy. Titan is very cold. It's out at 10 AU, so it's very far from the sun, a lot cooler than you'd find on Earth, and in fact, its surface temperature is about 200 degrees below the freezing point of water. So water on Titan is not really water anymore, it's ice, and ice is a rock. However, just like it's unlikely to see solid rock on Earth flowing across the surface, occasionally you do see that. If you've ever been to Hawaii, you have seen the lava there, which is liquid rock flowing across the surface. And similar things can happen on Titan. In impact melts, on the left here you see Sinlap, which is one of the craters we found on Titan. And then on the right here is possibly a cryovolcanic flow. So it's possible that you can get liquid water for short amounts of time in these impact melts and cryovolcanic flows. And these are doing thermal conduction modeling. We expect them to probably last somewhere between 100 to 10,000 years. So not a lot of time compared to the scale of the solar system, but maybe enough time to do interesting chemistry. And that's what we're investigating here in this work. So next slide. So the question is, given a freezing time scale of 100 to 10,000 years on Titan, is there enough time to have interesting reactions between these Titan aerosol organics and water in these transient liquid water environments? In addition, we want to consider what these reactions might tell us about possible origins of life scenario on the early Earth. And one good analog might actually be the cold origin of life hypothesis, which I'll just mention here briefly. Go to the next slide. So as I touched on briefly earlier, when life began, presumably about 4 billion years ago, the sun was a lot fainter than it is now, and the Earth was correspondingly, would probably have been cooler had the atmosphere been the same. So it's possible that there might have been more ice on the Earth. It might have been cooler, and so we might have had life starting in basically little half-frozen ponds. And this might actually have protected early life. Ice is good at protecting life from, say, solar UV, also perhaps the increased amount of impacts that would have been around at the time of early life. So if you go to the next slide, there's a little schematic showing how life might have started in this kind of cold origin life scenario in a small amount of cool water under an ice layer, which is protecting it from impacts and UV. So we're thinking here about how the reactions that may be taking place in these transient liquid water environments on Titan might inform us about the origin of life on Earth. So go to the next slide. So here's the plan. We want to find out how much of these oxygen-containing prebiotic biomolecules we might find in aqueous solutions. And so number one, we're going to make our organics in a manner I'll describe in a minute, which I'll call tholins probably throughout this talk. Next, and then we're going to figure out how fast these organics react with the organic polymers at different temperatures and in different solutions. And then lastly, we're going to compare these timescales to timescales for which we'd imagine liquid water might be available on planetary surfaces. So if we can go to the next slide. So I'm going to study all the possible reaction pathways. I'm going to do two studies. The first one is just in pure water at temperatures ranging from its freezing point, 0 degrees Celsius, up to about 40 degrees Celsius. And this is just to get that first cut at how fast these reactions take place. And this applies both to Titan and the early Earth. I also want to do a separate study that only applies to Titan because it's very likely that any solution that we find on Titan would probably contain quite a bit of ammonia. It's predicted to be present in, say, a subsurface ocean, not unlike the one we expect also on Europa. That to have a subsurface ocean on Titan, which there is evidence of, that we'd need ammonia to have that there. So we're going to start first with the water study, so it's just in pure water. And so the first step, as I mentioned earlier, is we want to make our organic molecules. And so can you see it if I move my little arrow around?
No, you don't have an arrow.
Okay, let me get an arrow then.
No, I'm running your slides.
Oh, all right. I don't have an arrow. Okay, all right. This will be a thought experiment then. If you see the U-tube on the left, so what happens there is that gas comes in on the left, and it flows through that U-tube, and as it's flowing through, it's sparked by the two electrodes you see there, and this dissociates the gas, the methane and the nitrogen, and then they recombine to make these heavier organic molecules, which attach on the sides of the U-tube, which you can see labeled there. And then we can extract it, and it looks sort of like this brownish, orangish, reddish sort of substance, which you can see on the right there. So that's how we make our organic molecules. And the next slide. And so then the next step is that we're going to put them in water. So on the left, you see that's our apparatus for making the tholins, and then we dunk them in water. Obviously not that water, but just that represents water. And then place them in a very high-resolution mass spectrometer. And what's really important about this work, and this is actually the key to the entire work, is that we have this really high-resolution mass spectrometry. What it allows us to do is tells us exactly what molecule we're looking at. It's so good, in fact, that we can tell, say, an oxygen molecule, an oxygen atom from, say, CH4, or a nitrogen atom from CH2. So this is really important when we're trying to look at these tiny differences in mass, that we're able to have such high mass resolution. So next slide. And here's an example of one of our mass spectra. And you can see there's a whole bunch of different molecules in this spectra. On the x-axis is the mass. And you see we get so those little peaks there at every mass unit. Those peaks actually, we'll zoom in a minute, and you'll see there's actually quite a few peaks inside of those big peaks. And then on the y-axis, we have intensity of the molecule, and that basically gives us tells us a little bit about how much of that molecule we have, so we can monitor that over time to see if that molecule is growing in intensity or decaying. And I'll draw your attention to the peak on the very left-hand side, which is labeled as an internal standard. And so we have internal standards which are presumably remain constant throughout the run, so then we can compare them to our other molecules to see whether or not they're decaying or growing. And if you go to the next slide, you'll notice that after 6 hours at 40 degrees centigrade, these three non-oxygenated molecules I've highlighted here in the middle are now about the same level as the internal standard, whereas before they were higher. And then if we go to 25 hours in the next slide, you'll see that they've actually dropped. So this doesn't mean that the internal standard is growing. What it means is that these organic molecules are decaying. Presumably, they're reacting with something to create a new molecule. So if we go to the next slide, I'm going to zoom in on this one peak at 251 mass units, and we'll zoom in and we'll see what's going on there. There we go. And so we can see that we have an oxygenated molecule. This is, again, at 0 hours. So this initial peak is probably just contamination that takes place when we're making the organic molecules. Unfortunately, some air leaks in on this very oxygenated planet we have here. But you'll notice that over time, if we go to the next slide, it's actually grown compared to the non-oxygenated species next to it. And then after 25 hours, it simply dwarfs the entire spectrum. If we can go to the next slide. So we go. So here, for example, is an example of an oxygenated species that is growing, and so we can look at that on the next slide at 4 different temperatures, and they all show this beautiful what's known as the first-order growth curve. Basically, you'll see the equation up at the top right. That equation just comes as a result where we assume the concentration of the molecule is directly proportional to the rate at which it's growing. So we see that these nice growth curves at all 4 temperatures that we looked at. And you'll notice also that the growth rate at 0 degrees Celsius is a lot lower than the one at 40 degrees Celsius, which is something else that we'd expect. We'd expect reactions to slow down at lower temperatures. So if we can go to the next slide. We can see that we did this for many different... Ooh, I can hear myself. Anyways, so we did this for many different molecules at these 4 different temperatures, and we'll notice that even at 0 degrees Celsius, so that's a pretty cool solution, the growth curves had half-lives on the order of days. So what this means is that the time it takes to go halfway between the starting point and that plateau point is on the order of days. So given that we have at least 100 years in, say, an impact melt pool or cryolava on Titan, and probably much more than that on the early Earth, it seems like these reactions are happening very quickly with comparison to the timescales for which water is available on planetary surfaces. So that was very exciting. So if we go to the next slide. So now we're going to talk just about a Titan-specific experiment I did. Basically identical to the one I just talked about, but now we did it in ammonia water, which is relevant for Titan. So instead of placing them in pure water, we just placed them in 13 weight % ammonia water. And what's great about this is we can do even lower temperatures, since ammonia water freezes at a lot lower temperature than pure water. We could take it down to temperatures as low as minus 20 degrees Celsius.
No battery, your battery shut off.
Yeah, our computer just shut off too. So I was trying to indicate to somebody to plug it in, but I guess my miming didn't work.
You can just switch to that other one that you had and tell me when to go to the next slide.
Oh, super. Yay, and now I can control it myself, perhaps. Can you see it now?
No, just tell me next slide.
Okay, next slide. So can you see the spectrum there now?
Yes.
Okay, great. So here is a very similar spectrum to the one I showed you in pure water, except this was done in ammonia water. And we're going to again zoom in on one of these peaks. So next slide. And then, and so again, here we have a very similar spectrum to the one I showed you before. We have a tiny contamination of oxygen there and a very big peak without oxygen in it. But then as time progresses, next slide, the oxygenated peak again begins to grow. Next slide. And after 50 hours at room temperature, it's quite a bit higher than every other peak at that same mass. So, and we can again plot a growth curve like we did in water. Next slide. And we can see that it follows the same pattern as we saw with the water. And if we go to the next slide, we see that we did this again at these very lower temperatures, as low as minus 20 degrees centigrade, where this is still liquid. And even there, it reacted on the order of days. So even in these really low temperatures in ammonia water, the reactions are still taking place on a very fast time scale. So if we can go to the next slide, we can show you, here we go, perfect, that this is true of all cases at all temperatures, that in ammonia water as well as water, that these reactions are occurring on timescales of days. So if we go to the next slide. So then the next question we want to ask ourselves is, well, how does this, is ammonia any better than water? You know, is Titan going to be a better place to, say, start making life than Titan because it has ammonia? And so we can compare, we can make direct comparisons at two temperatures, at 0 degrees Celsius and room temperature, because we did both of those temperatures in both studies. And we see here in this graph, we've got rate on the y-axis and then mass on the x-axis, and then the blue dots represent those rates done in ammonia water, and the red dots represent those rates done in pure water. And you'll notice just by looking at this graph that the ammonia water, in general, has higher rates at the same masses than in water. And if we do this again at room temperature on the next slide, we'll see the trend is actually even better at room temperature. We have 70% of these masses do not overlap in rate, and of these, 90% are higher in ammonia water. So it seems, what we seem to be seeing is that rates are faster in ammonia water, which is great because when you're down at temperatures like minus 20 degrees centigrade, you really want faster rates. So if we go to the next slide, kind of just ponder about why this might be the case. And it might simply be an effect of pH. What we've done is, in essence, by going from pure water to 13% ammonia water, is that we've increased the pH from about 7 to about 11. And there was some studies done in the 60s where they looked at amines, and that's basically any organic group attached to an NH2, which are almost certainly a part of the organic compounds that we're looking at. And you'll notice that over the pH range of interest, rates either seem to rise or stay about constant. So it could simply be due to the fact that the pH is increased. However, another more exciting possibility is there actually might be new reaction pathways being introduced by the presence of ammonia. And this is borne out by observation. Here you can see we actually did a study in labeled ammonia water where the ammonia was labeled with nitrogen-15. And so you can see, again, we have growing oxygenated species. You can see that the species I've highlighted on the left is growing, as we saw in every other study. But then, right next to that big peak on the right there, you'll see a species that contains absolutely no oxygen, but it does have a nitrogen in it, N15, and it too is growing. So it looks like, in addition to just having an increased pH, we are also getting a new chemistry made possible by the presence of ammonia, which we see here in this labeled study. So just to wrap up, we can go to the next slide. The take-home message from this talk is that if you take these sort of haze particles that we might find on Titan and perhaps also the early Earth and place them in liquid water, you can very easily, on the order of days, produce oxygenated species. And so it's important, this is important to start the steps towards biology by incorporating oxygen into these species. And so it seems to happen quickly and easily. So that's the take-home message. And I'd just like to thank my co-authors and my advisor, Dr. Levine, and my funding agencies, and you all for your attention. Thanks. Great. Thank you. Yeah.
Okay, in the interest of time, we're going to take just one question while I switch presenters over to Billy at University of Washington. So is there anyone that has a question they'd like to ask? And we will go back to questions at the end of Billy's talk as well. Looks like Ames has raised their hand. So Dave DeMarais, let's go to your site there.
Yeah, just the question, and that is the specific molecules that you see growing. Are there any additional comments you can make about what these species are?
Right.
The nitrogen heterocycles?
Yeah, that was actually one of the reasons we started this work, was hoping to get some information on structure about these molecules. And so one of the things we looked at was looked for correlations between growing oxygen species and decaying non-oxygen species, because then you could perhaps link them to see what reaction might have made these molecules. And unfortunately, there was no clear consensus. We saw some evidence of loss of, like, say, ammonia or methylamine or ethylamine, which might indicate that we had alcohols or aldehydes being made. And this, we also did a fragmentation study where you basically bombard the molecule and they split apart into little molecules. And we did see evidence of both water and carbon monoxide coming off of these in these fragments. So there is some evidence that perhaps we're seeing sort of imine hydrolysis, but I wouldn't say I wouldn't say it's a closed book. I think there's a lot of still uncertainties about what exactly it is we're dealing with, unfortunately. So.
Great, thanks, David. Goddard, I know you have a question, but if you can hold it till the end so we can make sure that Billy gets his time, and then we'll open the floor to questions and discussion at that point. So, Billy, it looks like your slides are working, so go ahead, take it away.
Okay, great. Can everybody hear me?
Yes.
Can you hear me? All right, thanks. Thanks a lot. Thanks, Catherine, for getting people thinking about chemistry, because I'm a biologist. I'm supposed to talk about prebiotic chemistry. The reason why that's relevant is because the organisms that I study live in a place that's dominated by this chemical reaction called serpentinization. And I'll talk a lot about serpentinization in just a moment. First, I'm going to give you some like the overall big picture astrobiological themes of why related to this talk. So first of all, this serpentinization reaction produces lots of methane, and some people have proposed that the methane that's been detected in the atmosphere of Mars might have been produced by this process. The study site that I have, which I'll explain in a few minutes, some people have said might be a great place for the first metabolic reactions to evolve. And also I study biofilms, which are a community of cells, and some people think that the first cells might have evolved from a community of cells like the ones that I study. So those are just the big picture themes that I want you to keep your eye out for as I talk about the details of my thesis project. So this is serpentinization. It's the chemical reaction that occurs when rocks, pretty tight rocks like olivine, come into contact with water, and then they turn into this black rock called serpentinite. And the reason why this is biologically important and important for the origin of life is that when this reaction happens, first of all, it's exothermic, which means it releases lots of energy, which can mean heat, and that drives hydrothermal circulation. There's also lots of hydrogen that is produced, so hydrogen is a great energy molecule for living things. It also produces organics, so it's food for biology. And we don't know all the other things that might happen when this reaction occurs. For example, not much is known about what happens with nitrogen and sulfur species during this process. But when thinking about the origin of life, you want a chemical reaction that happens spontaneously between rocks and water that produces energy and food for life. That's a pretty attractive process if you want to think about the origin of life. So I study a place called the Lost City hydrothermal field. It's on the Mid-Atlantic Ridge, although unlike most hydrothermal fields, it is not right on the spreading ridge. It's actually 15 kilometers away. And the reason why it can do that is because it's not actually a volcano. It's not primarily driven by magmatic heat like most hydrothermal systems. Instead, it's driven by the serpentinization reaction. The serpentinization reaction occurs in the subsurface, it produces heat, and it drives fluid flow in the system. And here I often show a movie that shows you the fluid coming out of the chimneys and the biofilms that are living on the chimneys. The movie is not going to work today, but here's a picture of a chimney at Lost City. First thing you might notice is that it's bright white, and that's because it's made of carbonate minerals, not metal sulfides like black smokers are. The chimneys at Lost City never fluid that's much more than 100 degrees Celsius, whereas the black smokers that you see on the Discovery Channel and so on, they can get 350 or 400 degrees Celsius. So it's much cooler. The pH is very high, up to 10 or 11. So it's very high pH. It's like toilet bowl cleaner. And as I said, lots of hydrogen and methane are coming out of these chimneys. And also, there are no tube worms. There are no gigantic animals like you see on black smokers. Hardly any animals at all. Hardly any large animals, anyway. There are lots of microscopic animals like tiny arthropods and gastropods. But the most obvious sign of life at lost city are actually microbes, because they form these thick, snotty biofilms that are covering the chimney. You can't really see in this picture. But if you look at these chimneys, they're just completely, every single available surface area is coated with a thick snot, which are biofilms. As I said, lots of organics come out of these chimneys, and Gyura Proskourovsky, a colleague of mine at Woods Hole, has studied these. And here's a plot showing the concentration of these organics, up to 4 carbon atoms per molecule. And he has some stable isotope evidence that indicates that these were actually synthesized abiotically on a subsurface of lost city. These organics are not just byproducts of modern life. And another group has done some modeling on lost city chimneys. It shows that because they're so porous, it's a great way to concentrate organic compounds that are coming out of the fluid and put them into close proximity with each other to build larger organic compounds. That's really important for thinking about synthesizing things abiotically. So here's some of the details on the chemistry of the lost city fluids. As I said, there's lots of hydrogen, millions of times higher than you'd find in normal seawater. There's really a lot of hydrogen here. There's a significant amount of methane as well. Because of the high pH, there's almost no CO2 available for microbes. It's all either precipitated out in these carbonate minerals. But there are other simple carbon compounds that we don't normally think of with most organisms. Formate and acetate are actually more biologically available than CO2. And I'm going to talk about the significance of that in a bit. There's a little bit of fixed nitrogen in the form of ammonia and nitrate, but really N2 is the best nitrogen source. So as a biologist, I look at this chemistry and think about, if I were to imagine the ideal organism that could survive in this environment, what I would think of is something that could use hydrogen as an energy source, of course, and something that could use a carbon source other than CO2, so like methane, formate, or acetate. And if it could fix nitrogen, that would be ideal. So when we looked at these carbonate chimneys under the microscope and we extracted DNA, what we found is that the hottest interior-most portions of the chimneys are completely dominated by this one organism that belongs to the Methanosar snaeles group of methanogens. And that's interesting because the Methanosar snaeles group of methanogens could fit this criteria of the ideal lost city microbe. Some members of this group can utilize hydrogen with CO2, but they can also use other carbon compounds like acetate and other methylated compounds. They can even use methane and oxidize the methane instead of produce it, and some of them can fix nitrogen. So no one species in this group can do all of these things. So no known member could be the ideal lost city microbe, but perhaps this one at Lost City can do all of these things. So there are other things besides this one methanogen that live, but they mostly live in the more oxidized, cooler portions of the outsides of the chimneys. What I'm going to be talking most about in this talk is the hot, really interior portion of the chimney, where there's really just this one group of methanogens that dominates. And so for one aspect of my thesis is to test the hypothesis that these methanogens can utilize hydrogen and they can use carbon compounds other than CO2. So like a lot of organisms in these types of environments, we can't culture the things, unfortunately. I've tried very hard. So what I did do to try to get an idea of what these things are actually doing on the seafloor is I just took a chunk of the carbonate chimney and incubated it at high temperature and high pH and used some isotopically labeled compounds, a little bit like what Catherine was doing, to see what happens. So here are two experiments. On the left, I tested whether isotopically labeled CO2 was converted to methane, and the second one was whether isotopically labeled methane was converted to CO2. And I also tested whether either of these processes was stimulated, did it go faster in the presence of hydrogen. And I'm not going to look at the numbers in too much detail here, but it's obvious that when I added hydrogen, both of the reactions went faster, which is not what you would expect if methane production and methane oxidation were competing or were reverse reactions compared to each other. In that case, hydrogen would have stimulated to go in one direction but not the other. But in this case, both directions of this reaction are going faster, which is a bit surprising. And what it must mean is that they're not reverse reactions, is that there must be, that these methane production, methane oxidation must be two different parts in the same overall pathway, and they're somehow linked, they're somehow cooperating. And we want to figure out how that's happening. So one clue is to look at the thermodynamics of the chemistry at low acidity. So as I said, CO2 is not a very good source because it's not very abundant. In this table, I've listed the energies of reaction for the reactions associated with using these different carbon compounds. So here's CO2, here's acetate, here's formate, and methane. And all these reactions are favorable under these conditions, 90 degrees Celsius, pH 10, with lots of hydrogen and methane around. But the amount of energy you can get from each of these reactions, given how much of that substrate is coming out of the chimney per unit of time, is very different. CO2, it would take you 500,000 years to get the amount of biomass we see at low acidity if you only use CO2 as your carbon source. So that's not very good. Whereas if you use acetate, formate, or methane, you could do it in a very small amount of time. So clearly, they must be using, just using the chemistry, they must be using acetate or formate or methane instead of CO2. So we have a little bit of metagenomic DNA sequencing data from these low acidity chimneys. And one of the things that we found is a gene that encodes the enzyme acetyl-CoA synthetase. And this is a gene that is only found in organisms that directly use acetate from the environment. And the closest relative of the version that we found at low acidity are genes that belong to acetate using methanogen, make methane from acetate. So this is really in support of our hypothesis that these things are using acetate instead of CO2. At least they are able to. But this is confusing because we don't know of any methanogens, that I know of anyway, that can use both hydrogen and acetate. Usually if you have acetate, then use it. If you have hydrogen, then use that. But there are no methanogens that I know of that use both of them at the same time. But at low acidity, it would be really great if you could use both, because it would be crazy not to use all that hydrogen, and acetate seems like a good carbon source because there's no CO2. So here I have a diagram of how that might work. And what I'm thinking is that when organisms use acetate, or methane as well, and convert it into biomass, there's a little bit of CO2 that leaks out. And the CO2 that leaks out of that might be actually a better source of carbon dioxide than just getting it from the fluid. So perhaps some of that CO2 is derived from acetate and then passed over to react with hydrogen to make methane and as part of energy conservation and making ATP. So in that way, you'd actually be, if you use methane instead of acetate, you'd be oxidizing methane, you'd be making methane and getting ATP and making biomass at the same time. So this is the hypothesis I like to test. I'm not anywhere close to really proving that's what's going on, but that's what I'm thinking could work. And I think this has implications for origin of life studies because some people think that the acetyl-CoA pathway, this pathway that methanogens currently use to fix carbon dioxide into biomass, might have been the first carbon fixation pathway. And if it did, it probably evolved in an environment with lots of acetate. It means it's a lot easier to make acetate into acetyl-CoA, which is the first molecule in the acetyl-CoA pathway, than it is to make CO2. And if acetyl-CoA pathway evolved in a high pH environment, perhaps dominated by serpentinization, then obviously acetate would be the one you'd use, not carbon dioxide. So if you want to learn more about this, I highly recommend this review that recently came out in Nature Reviews in Microbiology, which talks a lot about that idea. So in our metagenomic data, we also have some insight into the nitrogen cycle. We found operon for encoding 4 genes involved in nitrogen fixation. So I mentioned earlier, one of my hypotheses is that this ideal microbe would be able to fix nitrogen because there's so little ammonium nitrate around. So here's 4 genes that are responsible for that. And if you look at the diversity of one of these genes, this NIFH gene, and the different low acidity chimneys, you see that there's several different versions of this NIFH gene. But more than that, slightly different hydrothermal chimneys have different NIFH genes. So that shows there's quite a bit of diversity in this nitrogen cycle. If you look at the nitrogen genes, much more diversity than you see if you just look at the 16S ribosomal RNA, which is usually that barcode that microbiologists use to assess species diversity. So it looks like there's just one methanogen that's living in these chimneys, but this one methanogen is accounting for lots of different NIFH genes, lots of different genes involved in nitrogen fixation. That's kind of confusing. And one possible explanation is that there's a lot of horizontal transfer going on between slightly different subspecies within this one methanogen species. And we also have some evidence for that, looking at our metagenomic data where we find lots of different transposases, which are enzymes that are involved in moving DNA around within a genome or between genomes. So here's a plot of the size of an assembled DNA fragment in our genomic studies and the read depth or coverage, which is a measure of how many times that particular DNA sequence was found in the whole data set. And we see this peak of highly abundant but very small DNA fragments, and almost all of them contain these transposases. And so this is indicating that there's probably a lot of plasmids or viruses in these biofilms. And what we want to know is what they're doing. They might be involved in moving these nitrogen fixation genes, which are known to be often horizontally transferred between cells, and actually creating more diversity, more metabolic diversity within this biofilm than we see by just looking at the typical barcode, 16S ribosomal RNA diversity. And so now I want to talk a little bit about biofilms as an astrobiological study topic. So biofilms are social communities. And some people have said that biofilms are multicellular microbes. And there's a modeling paper that recently came out in PNAS. And it shows that if you horizontal transfer of DNA, of genes within these biofilms, can actually introduce and stabilize the existence of multiple genotypes within that biofilm. And the reason why that's interesting is because it's known that lots of cells within biofilms can actually differentiate without really changing their genotype, but can change their phenotype to fill specific roles within that biofilm that are necessary for the overall health of the biofilm. So this is an example of basically microbes cooperating to fulfill different functions in order to have the overall multicellular entity have more evolutionary fitness. And that's a very interesting thing for thinking about evolution, and particularly thinking about perhaps how the very first cells evolved. There's this idea of the pre-Darwinian community of pre-cells, which is an idea that the very first cells evolved from primitive cell-like things that had very few genes. Each cell had very few genes. And therefore, they're metabolically dependent on each other. And they compensated by this very small genomes by transferring genes among each other rampantly, not really caring who it came from or where it was going to. And as this community evolved, the cells as we know them became more resistant to horizontal transfer and became the cells that we know. And so I'm thinking that studying biofilms like the one at last study that have lots of horizontal transfer going on, that have limited genomic diversity but somehow have more metabolic diversity, possibly as a result of this horizontal transfer, might be a great way to studying the dynamics of this hypothetical pre-Darwinian community of pre-cells. So I've been saying that if you look at this typical 16S ribosomal RNA barcode, that you really only see one species in the phenogens. And that's true with most techniques that we're now only scratching the surface of what the most abundant microbes are there. But recently we have been able to get this very nice data set from the International Census of Marine Microbes, which is using this new sequencing technology that can actually sample the environment in much, much greater detail, has a much higher detection sensitivity than we've had before. And it can detect very, very rare organisms. So here's a figure from one of our samples that was sequenced with this technology, where we got 20,000 sequencing reads. So 20,000 DNA sequences that came from this one sample. 85% of the 20,000 have the exact same DNA sequence that corresponds to this methanogen that dominates the biofilm. But that remaining 15% represented 87 additional species, or operational taxonomic units, OTUs, which is the microbiologist's fancy way of saying species. And if you look at how many of these rare species were detected with increased sampling depth, which is what this curve is showing, is that you found 88 species after sequencing almost 20,000 DNA sequences. But that curve isn't anywhere close to leveling off. So if we had a technology that could sequence even more than 20,000 DNA sequences from the environment, we could find even more species. And so this is bringing up the idea of the rare biosphere that you might have heard about before. And we see that even in an ecosystem where there's really just one species, at least we thought there was just one species with the previous technology, there's a rare biosphere within, as a background to that major species. And in this particular case, most of these 87 additional species are very, very closely related to this dominant species. And so we don't quite know what that means, but we're wondering if that might be a reflection of this metabolic diversity that we're seeing in other genes. And right now we're trying to figure out how to correlate this metabolic diversity and this rare 16S diversity. So I've been talking sort of abstractly about these things, but here's a picture of what this biofilms look like. This is a transmission electron micrograph. You've got your scale bar here for 1 micron. Basically, each one of these cells is approximately 1 micron wide. This gray part and the white part here, that's the carbonate mineral. So you see these biofilms are directly attached to the carbonate minerals. And these cells over here have this very characteristic morphology of the cells in this methanosarcinase methanogen path. Very characteristic. But there's lots of other morphologies here too that we were not expecting to see. And we don't really know what those things correspond to. Based on the 16S sequences, at least 85%, possibly more of these cells should be the exact same thing, but that's not really what we're seeing here. So this might be an example of how cells within a biofilm can differentiate to fill various roles, even if they have the same genotype. And that's, as I said before, that's hypothesized to be what might have happened in these very first community of pre-cells. So I'm just summarizing the results here. To reiterate, the chemistry of these lost city fluids, because their serpentinization is very high pH, fairly warm, about 100 degrees, lots of hydrogen and organics. We found both methane production and methane oxidation are being mediated by microbes, and they're stimulated by hydrogen. We have this metagenomic data that shows that they have the genetic potential to use carbon compounds other than carbon dioxide. There's lots of horizontal gene transfer that has the potential to be going on. The phylogenetic diversity, there's a rare biosphere, as evidenced by the 16S sequences, but there's also lots of diversity in the nitrogen fixation genes. And you see this great morphological diversity despite limited genetic diversity. All right, so here's my last slide. I'm just throwing out some ideas about how all of this is astrobiologically relevant. For example, if these cells have differentiated into specific niches, are they engaging in a centrifugal partnership? Are some of those cells that we saw in that picture undergoing methane production, some of them doing methane oxidation, and they're somehow cooperating with each other, passing along metabolites, which might be a great thing to do in a biofilm because it inhibits diffusion away from things. So, for example, if you've got CO2 from that acetate, instead of precipitating it out immediately, the biofilm might make that carbon dioxide aqueous and dissolve for a longer period of time than it would if it had been outside the biofilm. And then, if these centrifugal metabolic relationships were happening in a very early biofilm, early in evolution, perhaps that might have contributed to this unity of biochemistry that we see today. Just speculation. As far as extraterrestrial life, I think it's a good question to ask. Could you have an organism living entirely off of serpentinization? I think that might be happening at Lost City, but because there's so much oxygenated seawater circulating around, it's kind of hard to tell at Lost City. And also, if the methane on Mars is being produced by serpentinization, I think we got to look in the subsurface to see if there are biofilms like we see at Lost City directly living off of that serpentinization. Okay, I'll stop there and open up for questions.
Great. Thank you, Billy. All right. Well, I want to turn questions over to headquarters and thank Max Bernstein, Michael New, and Mary Wojtek for joining us. And I believe there's a question there. And you might be muted. So make sure you... yeah, go ahead.
Okay. So I just had a question about what you knew about sulfate reduction at the site.
No, she's asking me to.
Can you repeat the question, Mary?
Okay, my question was for Billy, and I was wondering if he wanted to comment on if he had any information about sulfate reduction, either from his metagenomic or any direct measurements at the velocity sites he's been looking at.
Good question. It's a very confusing topic. In the metagenomic data, that we did find one gene, this DSR gene, that's involved in sulfate reduction, but it's not involved. It belongs to a firmicute. It does not belong to the typical bacteria that are involved in anaerobic methane oxidation. In fact, we don't find any of the typical bacteria that are found associated with anaerobic methane oxidation. So we're still trying to figure that out, but it's unusual.
Sorry, I was just wondering if you thought it might be involved when you proposed syntrophic partnerships, and if it could have been a more traditional type of sulfate reducer that's involved in a syntrophic relationship facilitated by the supply of hydrogen with the methanogen.
Yeah, I think sulfate reduction could definitely be involved in this possible syntrophic relationship, but it's not involving one of the bacterial species that have been found in other environments. It has to be a different bacteria if it's involved. And when looking at the bacteria that are there, it's hard to imagine one of these bacteria living at 80 degrees or 90 degrees Celsius in a pH 10. But maybe it's happening. Looking into it.
Great, thanks. Okay, Goddard Space Flight Center. You have a question?
Let's see. Actually, two questions. One is a comment. Can you hear me?
We can hear you.
Okay. One is a comment to Billy, and that is that our team has just submitted two papers which are now currently under review regarding methane on Mars. And the second one reports detection of significant plumes over two regions of interest to your talk. One is over the southeast quadrant of a low-shield volcano, and that quadrant shows evidence of local collapse, which suggests that the surface is connected through fissures, at least seasonally as ice clears, to subsurface zones that we can't say whether serpentinization is still occurring there or not, but it's interesting to consider your remarks regarding biofilms in that connection, which would become a prime region for landed missions to further explore the effluent gases and their isotopic ratios. Do you have any further comments on what kinds of signatures might be sought in such regions to differentiate the suggestion you made, serpentinization plus biofilm generation, from other means of producing methane?
Yeah, I think it would be extremely difficult. If you look at the isotopic signatures of hydrogen and methane at Lost City, I brought up a slide here. I don't know if everyone can see it. But other authors have used this figure to show how you can differentiate between biogenic and abiogenic methane. If you look at the methane at Lost City, it has a pure abiogenic signal. And that could be because the methane being produced by the biofilms is very small compared to the methane being produced abiotically, or because the isotopic fractionation associated with these biofilms is extremely small due to the very high temperature. It's known that very high temperature reduces fractionation, or also because the biofilms are using substrates that are coming from this abiogenic source, and they're not fractionating it very much, and therefore you don't see it. So I guess my comment would be to be careful to be disappointed if we saw such a signature at Mars, because that's the signature we see at Lost City, and there's lots of biofilms there. So as a biologist, what I'd like to see is some microscope images, of course, but I would suggest using sulfur, like maybe sulfur isotopes might be a better way if sulfate reduction perhaps were involved.
Well, as you probably know, Barbara Sherwood-Lawler, particularly from the IPTI team, has taken the lead in showing that it's not sufficient to look for fractionation of carbon-13 in samples produced from to decide whether they're biogenic or not, but rather that plus looking for hydrogen fractionation in the form of deuterium is far more diagnostic. Has that been done in the case of Lost City?
Gero Proskorowski at Woods Hole has done hydrogen isotopes, and he has shown has done some of the chimneys at Lost City show evidence of hydrogen utilization. But however, the main chimney plume that we mostly study does not really show much evidence of hydrogen utilization, which we don't really understand why that is. But there's lots of biofilms in the chimney, so yeah, I mean, we definitely want to measure those gases, but we should be careful to be too disappointed if they don't show an obvious biological signal.
Okay, Estelle, I'll have another question for Catherine, but perhaps we should let this...
Okay, yeah, we're on the hour right now, so if people need to get going, I'll let you do that. And I just want to thank everyone for joining. Michael, we'll come back to you, but let's go to Ames and let them ask their question, and then we'll come back to Goddard.
Yeah, hi, this is Dave DeMarais here. The question I have for you relates to the pH of the actual environments of the biofilms. People studying the cedars in California found very low activity at pHs 11. And so I'm wondering, do you need a little bit of dilution of lower pH water for the most robust biofilms to develop? Can you comment a little bit about pH of the microenvironments of the ones that you studied with the genetics and the activity?
The only thing I really have data on is that even that the rates that I measured in these methane production oxidation experiments were the same at pH 8 or pH 9 or pH 10. They vary a little bit, but I don't really believe the numbers because of other errors involved in the experiment. So I don't really know. I don't have any data that actually do anything at pH 11. I only went up to 10. And also, we don't have a lot of really good data on the pH from Lost City because we really, at least on the micro scale, relevant to microbes, because we haven't really put a good micro sensor down there to see what the pH, where a biofilm is yet. So mostly I don't really know.
Thanks. Okay, just a reminder, if anyone has questions, to raise your hand in WebEx. And Mike, back to you for a question for Catherine.
Right. Catherine, your talk was extremely interesting. I was interested in why you chose that mass range from 240 to 260 AMU. I assume you have other major peak ranges in your overall spectrum.
We are limited to about 100 to about 400 AMU, just given the mass spectrometer we're using. It can't go much lower than 100. And after about 400, you don't really see very many tholin species anymore. So I just showed you a range. Actually, the one figure I showed that in purple, which I can't bring up now because I don't seem to have any control over these computers, but that showed more of the actual range we would have, which is from about 100 to, which unfortunately limits a lot of these smaller mass things where you might be interested. And it doesn't overlap with the mass spectrometer we have on Cassini either, which only goes up to 100 mass units. So hopefully one day when we go back to Titan, we can make some more comparisons between mass ranges that actually overlap.
Well, I was also curious, I noted that your nitrogen heterocycles, you had only units with 1 nitrogen, 6 units, or 6 nitrogens, or 7 nitrogens, and I was wondering what happened to the others.
Well, it seems the ones that react tend to be quite highly nitrogenated. We do see species with just a few nitrogens in them, but they just don't seem to be as reactive. It seems like the ones that react have more nitrogen in them.
But even if you have 6 and 7 nitrogens, I would have thought you might see 5 and 8.
Yeah, we do. I was just showing a couple of examples.
I see.
We have
Okay.
hundreds of species that are reacting. Yeah, I just wanted to show just a few of them.
And when you look at the other major mass groups in your 100 to 400 AMU range, can you use the occurrence of those mass groups to identify what the heterocyclic structure might be and so on?
Yeah, and unfortunately, that's something we've had a lot of trouble with. Though we can tell exactly what the molecule is, since these are such big, heavy molecules, there's thousands of different structures they could have. And so we were just trying to do it by linking products to reactants. That was the best thing we could do. And unfortunately, the results were not very unambiguous. So I'd say the jury's still out. People have been trying to figure out structured tholins for a very long time, and it's been very difficult to do.
Thanks. We have another question here. Mike?
Let's switch to John Peters real quick, and then we'll come back to you, Michael. John Peters?
I have just two quick questions, one for Catherine, and the question would be related to what you just talked about, the how much control you have trying to make some smaller molecular weight, what you could try to use to figure out some sort of a mechanism of the hydrolysis.
Well, we don't really need. It's not that we need smaller molecular weights to figure out what's happening. The heavy ones work just fine too. By example, by linking these products to possible reactants. So I guess, and then you can just see. I guess what's your? I just understand.
If you make
Sorry, go ahead.
it smaller, reduce the number of possibilities for structure isomers. So you start
Sure.
to focus.
You're probably not going to make a haze in that case. You're probably going to make a gaseous particle which gets sucked out of the machine. Also, you're less likely to find those on the surface of Titan because they'd be in the gas phase in the atmosphere. So we're more interested in these heavier molecules.
Okay.
So people have studied the gaseous products too.
For Bill, you mentioned possibility for hydrogenase activity. Do you have any information, any genes, anything identified, what type of hydrogenase enzymes, metal enzymes could be present?
I have found some genes that have their best blast hits are hydrogenases, but I haven't looked into them yet so much. I'm just getting started looking at that data. But yes, we definitely have some evidence of that.
Okay, thank you.
John. Thank you. Okay, and Goddard, back to you, Mike.
Can I go ahead? Hi, this is Mike Callahan at Guarded. Great talk for both speakers, and I have a question for Catherine. going back to structures again. For all your molecular formulas, have you calculated the number of double bond equivalents in your suite of molecules? And if so, also on your mass spec, your tandem mass spec, or your MSN, can you look at the fragmentation and at least get down to if these molecules are linear or cyclic molecules?
Are you asking about unsaturation or?
Yeah, I mean, how many double bonds? Because these look like probably linear.
Yeah, there's probably, actually it's probably both. That's the thing. They're such big molecules, you're probably getting a little bit of everything. And they do seem to be quite, unsaturated. Unfortunately, you can't do a lot of these standard techniques on tholins because there's just this big blobby mixture with everything in them. You can't tell anything from anything, so which is why we needed this really high-resolution mass spectrometry to at least distinguish the molecules, the different molecular formula from each other. But yeah, I mean, we can tell, I guess, how unsaturated the molecule is, and studies seem to indicate that there are linear portions of these molecules. Although I wouldn't rule out rings entirely.
But can you do? I mean, you can do mass fragmentation in your ICR, correct?
Yeah. Oh, yeah. We did that. Mm-hmm. Yeah, and we found fragments of like carbon monoxide and water and hydrogen cyanide.
Does it look like hydrocarbon fragmentation? Because if these are ring structures, they'd be harder to fragment, right? So does it look like these are branched molecules or linear, or it's just too hard to tell?
Yeah, but we saw fragments. We didn't see a lot of, CH2 or whatever fragments. It was mostly, we were looking more for the oxygenated fragments anyways. But I guess we didn't really see much hydrocarbon fragments. We saw water and CO and hydrogen cyanide, and that was about it. So.
Thank you. Great talk, guys.
Thanks. Anything else from Goddard?
No, I think that's it.
I have a question.
Okay.
Oh, wait a minute, there's one more.
All right.
Avi Mandel from Goddard. I had a question for Billy. I was wondering, back to the question of the possibility of serpentinization or biofilms on Mars, the conditions obviously would be very different than the ones at the bottom of the ocean, and the temperatures would be different. pH might be similar, but especially in terms of temperature or pressure, how do these processes, these biological processes specifically, not so much the serpentinization, but the biofilm pathways work under different temperature and pressure conditions?
Well, we have no idea. The short answer. Especially if these are novel metabolic pathways, then we absolutely have no idea. A lot of all other cases of anaerobic methane oxidation actually happen at very cold temperatures, like 4 degrees. So I don't think temperature would be a huge issue as long as it's above freezing. And pressure, no idea really. These are about 800 meters depth, so not a huge amount of pressure there. Otherwise, I think that these things are adapted to high pH and high temperature because that's where they have to live in order to get the hydrogen and methane, but I don't think it's necessary.
Okay, great. Thanks, Avi. Any other questions from any sites? Speak up. No? Okay, looks like we're dwindling. I want to thank everyone for attending and for staying, and especially thank Billy and Catherine for their talks and the people at their end that were helping. And reminder to tune in on November 3rd for Jennifer Eigenbrog and Dominique Gleason, who will be giving presentations on extreme life. So thanks, everyone. Bye-bye.